Now, to relate the amino acids with that of the bases, there has to be something in between that we can relate the two things. So, what that could be, that is what we say is genetic code. So, genetic code means ki whether it is A base relating to a particular amino acid, it is C, G, or T, or it is a sequence of the bases that is relating. So, it was Gamo, a physicist Gamo, who concluded that there is there has to be a sequence of 4 into 4 into 4, that is 64 codons, that is the 3 bases together, that will be signifying different types of amino acids. So, the codon is a triplet was given by Gamo, because if we choose only 2 bases in a sequence, that will be leading only to the formation of 16 amino acids, that will be relating only to 16 amino acids, and there are at least 20 amino acids that forms the protein in living organisms. So, the codons means the 3 set of bases, nucleotides, which code for a amino acid, that is always a triplet. Let's now see the salient features of genetic code. So, genetic code is a triplet. Triplet means there will be 3 bases that will be signifying one amino acid. So, for one amino acid there will be 3 bases. The codon codes only for one amino acid, hence it is unambiguous and specific. Unambiguous means it is non-confusing. Specific means it is only one of the codon, that is the triplet bases will be coding for one amino acid. Some amino acids are coded by more than one codon, hence it is called degenerate type. Then the codon is read in mRNA in a contagious fashion, there is no punctuation, there is no stop, there is no comma. Then the code is nearly universal, that is from bacteria to human being, each codon will be coding for the same amino acid. For example, UUU would always code for phenylalanine. There can be some exceptions here. AUG has dual functions, it is coding for methionine as well as it is coding for the initiator codon. Now, for translation, what more is needed is, it's not only a decoded message, but there has to be a machinery where the process will occur. So, for that there are ribosomes, and ribosomes are of 2 types. It could be the 70S type or it could be the 80S type. 70S is in prokaryotes, whereas 80S is in the eukaryotes. Now, a ribosome is further made up of 2 basic components, rRNAs and proteins. So, let us see, the 70S type is further made up of 50S and 30S 2 subunits, and the 50S subunit is further made up of 23S rRNA, 32 proteins, 5S RNA, and the 30S subunit is made up of 16S rRNA and 21 different types of proteins. Similarly, if it is eukaryotic ribosome, so it is further made up of 2 subunits. The larger subunit is called 60S type and the smaller subunit is 40S type. 60S type is further made up of 28S rRNA, 50 proteins, and further 5S rRNA and 5.8S rRNA. What does this mean? S means it is S का मतलब होता है Sedberg unit. Sedberg unit, and it is the sedimentation coefficient. Sedimentation coefficient. That means this S signifies the rate at which the ribosomes are sedimenting in a test tube when centrifugation is done. So, this way the 2 types of ribosomes, they are further made up of larger and smaller subunits. The smaller subunit further made up of rRNAs and proteins, and this will be serving as a machinery for protein synthesis. Now, another one more important thing that is needed during translation, the next step of expression of gene is an adapter molecule. Now, what is an adapter molecule? This molecule is the tRNA, the transfer RNA, that will be carrying an amino acid from the cytosol and leaving it up to the site of protein synthesis. So, it was Crick who mentioned this, that will be requiring some kind of a adapter molecule during the process of translation. So, it was Hawley who gave the structure of tRNA. Let us see how it looks like. It is basically a 3D structure, looks like a L-shape structure like this with two ends. It is RNA only, but it gets self-coiled and assumes a L shape in 3D. This is the 3' end, the 5' end, and in 2D this looks like a clover leaf like model. Clover leaf like model. It has got these kind of three ends, three loops, and there will be some hydrogen bond in between. This is there are complementary bases and they get bonded together. Now on this 3' end there will be always ACC bases where the amino acid will be attached. This is called the amino acid attachment site with this ACC, and from 5' to 3' is CCA. And on the 5' end there will be bonding. Then there is a D loop, there is a T psi U loop, and there is an anticodon site. This anticodon site will be the site where the anticodon are present. Suppose the codon is UUU, so the anticodon will be opposite to U. It has to be AAA, and amino acid corresponding to that AAA will be brought attached here at this particular end. So now let's start with the next process of gene expression. This is called the translation. Now what is translation? Translation means कि on this decoded mRNA there will be a protein synthesis that has to occur. For this there is the decoded message from the DNA. This is called messenger RNA. Then what else is required is a machinery called the rRNA with a smaller and larger subunit, and what else is needed is the charged tRNAs. Charged tRNAs means that the tRNAs, they are added up with the amino acid. They are bonded with the amino acid. They are carrying the amino acid, specific type of amino acid is attached to a specific tRNA, and there are 20 different kinds of tRNAs in the cytosol with 20 different types of amino acid. And it is pre to the translation that these charged tRNAs will be already there, and these tRNAs will be coming on this side, and the process of translation will be initiated. Now before the initiation of the process, the amino acids they are attached to the specific tRNA in the presence of a specific enzyme, amino-acyl tRNA synthase. So these kind of enzymes are specific because the amino acids are specific and the tRNA carrying them will be specific. So the first step initiation will be for the pre to this step will be called the charging of the tRNAs. So for this the amino acids will be, this is an energy consuming process, and the tRNAs will be charged with, added up with the amino acids. So the first step of translation is called initiation, and during this process there will be a messenger RNA, mRNA, which is the decoded message from the DNA. There will be the smaller subunit of rRNA that will act as the site and the factory for the protein synthesis, and the one of the charged tRNA that carries methionine, because this is the initiating codon, AUG start codon. So there will be a complementary anticodon, and the specific amino acid methionine will be carried by this tRNA, and it will be brought and that will be left here. So the process for initiation will be like this. The cell needs a messenger RNA, some initiation factors, smaller subunit of rRNA, and the first charged tRNA carrying the methionine. Now for elongation, the second step is elongation of protein elongation step. So for this there will be the larger subunit that will come and attach at the site. The initiation factors will be leaving from here, and the next charged tRNA that will be responsible for CCA amino acid, that is proline, will be brought here. So there will be another charged tRNA with GGU, and it will be carrying proline. This is brought in. Now this larger subunit basically is further made up of two subunits, and this one of this piece is called 23S rRNA. Now this 23R rRNA acts as a ribozyme. Now this is acting as a catalyst for the peptide bond formation. So this is not only a smaller subunit of this rRNA, but also acts as an enzyme. So this is one of the enzymes that carries RNA. In fact, this larger subunit has two sites. One is called the P site and another is called the A site. P means peptidyl transferase site and A means amino acyl site. So now as soon as this another charged tRNA comes at this particular A site, so automatically, because there will be two charged tRNAs together and there will be two amino acids, charged tRNAs and amino acids together, so these two amino acids will be bonded bonded together by a peptide bond. So as soon as the A site is filled, the two amino acids will be linked automatically to the peptide bond, and the enzyme acting as a catalyst will be the peptidyl transferase enzyme, ribozyme, and the whole system shifts, the whole this will be shifting one step ahead. That means it will be like this. So the next will be CAG, like this. So now this will become the P site and the next will be the empty will be the A site. And as soon as this gets empty, there will be another charged tRNA that will be carrying the third amino acid with the anticodon of GUC will be brought here. And the process continues till this UAA kind of codon is reached, which is called the stop codon. The process looks something like this. So the smaller subunit, the larger subunit, the tRNAs, they are coming here at the empty site and the whole thing moves one step ahead. So the final step called the termination will be occurring as soon as there will be some UAA kind of UAA, UAG and UGA type of codons that will be encountered. So because there is no tRNA responsible to carry any amino acid for these three codons, so the process will automatically stop. And there will be some release factors that will be stopping the process. Now apart from this, there are some untranslated regions. Untranslated regions means there will be more codons than the required ones, and these untranslated regions, they are present at both 5' and the 3' end, and they help in the efficient translation process. Now when we talk about the expression of a gene, then we know that the process of expression has to be regulated, and the regulation is done by at 3 steps. It could be at the transitional level, it could be at the splicing level, or it could be at the transitional level. So let us take an example how the genes they are controlled, or how the genes the process is regulated. So for this we'll start with the lac operon. Now lac operon was first given by Jacob and Monod. Operon means it is an arrangement of genes that will be helping in the regulation of gene. So there are various kind of operons in all the organisms, in every cell, and they will be regulating the process of gene expression. Suppose there is an enzyme to be formed in the intestinal cells, so which gene will be coding for that has to form an mRNA and has to be translated. So there has to be a specific way for that. So let's talk about the lac operon. Lac operon means how the lactose hydrolysis is process of hydrolysis of lactose is regulated in E. coli. So it was studied by Jacob and Monod, and how the arrangement of genes are there. Let us see. It's a polycistronic way of arrangement. Polycistronic means there will be many genes controlled by a single regulator gene. So there will be the arrangement will be something like this. There will be structural genes ZYA. These 3 genes will be producing 3 different types of enzymes, and all 3 will be required for hydrolysis of lactose. And along with this, there will be an operator gene, promoter gene, and a regulator gene, and they have to be in this sequence: regulator, promoter, operator, structural gene. Now what is regulator gene? This I gene is for this is I word, I alphabet comes from the inhibitor word. Inhibitor means it produces a protein called the repressor protein, and this repressor protein will be will be physically binding with the operator region. Now what are these functions? This regulator gene, which is represented by I, this has to produce a repressor protein. The promoter gene, which is lying next to this will be the place where the RNA polymerase comes and physically binds, and the operator is the place where the repressor can come and physically bind. And then lies the structural gene. Now suppose the bacteria is not fed upon is not consuming lactose, or it is not given lactose in the medium, so it will be requiring glucose as such, or it has glucose for production of energy. But suppose there is no need of these enzymes to be formed, so how the process will be? We say the gene is in the off condition. So if the gene is in the off condition, so what will be happening? So when the gene is in the off condition, this I, a regulator gene, the inhibitor gene, will produce a repressor mRNA, a repressor protein that goes and physically binds at the operator gene. And because यहाँ पे RNA polymerase cannot now move towards the structural gene and cannot lead to our mRNA synthesis, the process is stopped. So there will be no enzyme synthesis, there is no wastage of raw material and energy if there is no lactose in the surrounding. But suppose sometimes the bacteria has to hydrolyze lactose. Suppose it is given lactose in the medium. So what will happen that day? So this repressor protein will be as such produced. But when you are giving lactose in the surrounding, so this lactose will be attaching with the repressor protein, and it will be changing the conformation. The protein is changed. And now because the protein structure is changed, so this cannot go and physically bind with the operator. And because it cannot go and physically bind, so RNA polymerase, which will be attaching here at the promoter region, can now move forward and can lead to the formation of mRNA, and the three enzymes, that is translation, will occur, and these three enzymes will be able to hydrolyze lactose, convert it into glucose and galactose. So the bacteria will be able to digest and consume lactose. Now let's talk about Human Genome Project. Now this was a project to decode the human DNA, to sequence the human DNA and decode every sequence. Now this was a mega project. Mega means that it was having a mega magnitude, and the requirements were also very large. Now what was the aim of this genome project? The aim was that there will be enormous data that will be generated. They will be needing a computational device for that, for storing the data and retrieval analysis. And what was the magnitude? Because the human genome contains 3 × 10⁹ base pairs, so the cost of sequencing for this total human genes were 9 billion. So it was a big project, and that was initiation was done by US Department of Energy, NIH, National Institute of Health, and the Wellcome Trust UK, which later on joined this. So what were the goals of this human genome project? This was to identify all the genes that are present in the humans. There were around 25,000 genes. Then to sequence all the 3 million base pairs, then store the information, improve tools for data analysis, and transfer the related technologies, and also address the ethical, legal, and social issues that will be arising from. What was the significance of this human genome project? Now suppose if we can decode the genes or all the blueprints of human life, so we could diagnose the disease and disorders of humans. We can learn about non-human organisms also, and we can understand their natural capabilities towards healthcare, agriculture, energy production, environmental remediation, and other things. So there was not only human beings, but there were other organisms like the Caenorhabditis elegans, so roundworm, and Drosophila, the fruit fly, plants like rice and Arabidopsis. They were also decoded. What were the methods employed? There were two methods. One was called expressed sequence tags, ESTs, and the other was called sequence annotation. What was expressed sequence tag? This could be your 1-mark question. To identify all the genes that expressed as RNA, and that was called expressed sequence tags. And the other was that when the genes only identifying the genes which are expressing themselves, and that was called sequence annotation. Now the method employed: isolating DNA, then digestion of the DNA, that was cutting it into small fragments, then amplifying the genes that were cut into fragments by using BACs and YACs. BAC is the bacterial artificial chromosome and yeast artificial chromosomes. And then the sequencing by automated DNA sequencers. They were developed on the Sanger's method of sequencing proteins. And then the arranging on the basis of the overlapping regions, and finally automated sequencer were able to sequence the whole of DNA. Now, what was the finding of this human genome when it was decoded? So there were many findings. It's a kind of data that you need to learn. The human genome contains 3164.7 million nucleotide bases, and an average gene consists of around 3000 bases, but the size varied greatly. The largest gene was of dystrophin, muscular dystrophin, 2.4 million bases. The total number of genes was around 30,000, but it was expected it has to be somewhere 1.5 lakh around. And almost 99.9% of the nucleotide bases were exactly same in all human beings. Then the functions of more than 50% of them, they are still not known. Then there are some repeated sequences, and these are the stretches of DNA sequences which are repeated many times, and they do not have any direct coding functions. Then chromosome 1 has the maximum genes, 2968 genes, and the Y has the fewest, 231. And the scientists, they could identify around 1.4 million locations where single base DNA differences, SNP, single nucleotide polymorphism, or they are pronounced as SNPs, occurred in humans. Now, one of the significance of decoding human genome was that we could go for DNA fingerprinting. Now, what is DNA fingerprinting? That the repetitive sequences which we have, or the DNA of two persons, cannot be same. They will be having certain sequence, they will be having certain differences in the DNA. So the process involved isolation of DNA, then digestion of the DNA by restriction endonucleases, separation of this DNA by agarose gel electrophoresis, transferring or blotting of separated DNA fragments on a nylon sheet, hybridization using labeled VNTR probes, and finally hybridization of this DNA fragments by autoradiography. Now, this whole process of decoding of DNA and DNA fingerprinting requires some small sequences which are different in all of us, and these are called the repetitive DNA. So, because they are small stretch of DNA repeated many times, they are called repetitive DNA, and they are separated from the bulk DNA by different peaks during density gradient centrifugation, and the smaller peaks that are obtained are called the satellite DNA. Now, these are rich in A or T, G or C, and based on that they could be micro satellite or mini satellites. So there could be some small definition questions, and these are the ones. Now, significance of DNA fingerprinting is that it could be used in the forensic science for determining the parent and for solving the parental issues, and in also studying the population and genetic diversities.